Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Nurainna Fatiha binti Izzuwan and I'm a third year student of Bioinformatics and I was supervised under Dr. Zuraini Alisha. Did you know that more Malaysian children are being diagnosed with autism spectrum disorder or also known as ASD each year? According to the chairman of National Autism Society of Malaysia, Felina SY Muhammad Faizul, by analyzing the national gross birth rate per year, there are around 8,000 to 9,000 born yearly that may have autism in Malaysia. However, there is no official registry for the number of individuals diagnosed with autism in Malaysia. Meanwhile, World Health Organization (WHO) estimates that 1 in 160 children has ASD, and its prevalence appears to be increasing globally. You might be wondering what are these facts has to do with my presentation. Hence, I would like to introduce my research title, which is Classification of Autism Gene Expression Data Using Machine Learning Methods. So here is the outline for my presentation, where it is divided into 5 parts, which are introduction, literature review, research methodology, research design and implementation, and lastly will be conclusion. For the first part, which is introduction, I'll give a brief explanation about the domain in this study, which is ASD, gene expression data, and I will also state the aim and objectives. ASD is a lifelong neurodevelopmental disorder that could affect human communication and behavior as it is related to social interaction, speech, and communication. This disorder makes it hard for them to relate to the outside world, and it is also hard for them to communicate with others. Aggressive and or self-injurious behavior may be present in some cases. Gene expression data is a set of data that holds significant information for the gene expression values of genes that are computed from the microarray technologies. The dataset is extracted from the gene expression process where DNA is converted into a functional gene product. A lot of research has been conducted using various machine learning algorithms to study ASD. Gene expression in ASD is used not only in an attempt to separate ASD into different subtypes, it is also used to select the most attributed genes. However, the uninformative gene is also included in the dataset due to the high dimensionality of gene expression data. Regarding this problem, this research is conducted to classify the genes into autism and non-autism from the gene expression data using machine learning methods. There are three objectives for this research, which are: first, to identify the classification method that can be implemented on autism gene expression data. Second, to implement and modify the classifier parameters that will be able to classify more accurate on autism gene expression data. Lastly, to evaluate the classification method implemented on autism gene expression data using accuracy performance. The second part is literally about the summary of literature review, in which a short explanation will be given for each machine learning methods used in this study. Classification plays an important role in organizing data. In this study, classification technique works by predicting the target class for each group of data, which are autism and non-autism. As for machine learning, it is a method of data analysis based on the idea that systems can learn from data, identify patterns, and make decisions. There are four machine learning methods used in this study, such as multilayer perceptron, k-nearest neighbor, naive Bayes, and support vector machine. The first method is multilayer perceptron. It is known as feed-forward neural network that use backpropagation for classification. It also has more stacked layers in which every layer consists of small pieces called neurons. The structure of MLP consists of three main layers, which are input layer, hidden layer, and output layer. Those neurons get input data X in the input layer and then share that data to the next layer. In the next layer, which is a hidden layer, each neuron will receive a weighted sum of the neuron output from the previous layer. In order to control the input, the activation function is implemented within each neuron. Next, k-nearest neighbor, in which the basic concept is to predict the target label by finding the neighbor class closest to them. The implementation of k and n is based on two operators, which are the value of k that refers to the number of neighbors to consider and the distance measure used. The significant part in using k and n method is choosing the right number of neighbors. It is essential in order to maintain the ability of the algorithms to make a classification accurately. The third method is naive Bayes. Naive Bayes is a supervised machine learning algorithms that use Bayes' theorem, which assumes that features are statistically independent. The classifier is assumed to have no relation to other classes with other features in the class. Naive Bayes has the advantage of being probabilistic models, resilient to missing values and real data noise. Lastly, it is a port vector machine which classifies the data points by finding n-dimensional hyperplane where n denotes the number of available features. A hyperplane is a decision boundary used to separate space into input variable. SVM can exist in two categories such as nonlinear SVM and linear SVM. Nonlinear SVM is used for dataset that cannot be categorized using a straight line, while linear SVM is used for data that can be separated linearly. SVM is widely used in classification of ASD due to its ability to provide a decent accuracy level in classification for high-dimensional data. A variety of studies in classification of ASD gene expression data are available in both machine learning and deep learning. However, the number of studies conducted in deep learning using gene expression data for classification of ASD is much more smaller than machine learning. Hence, machine learning is chosen to be used in this research. The third part in this presentation is research methodology, which consists of research workflow and performance measurement. There are three phases available in the research workflow. Phase 1 is about research planning and data preparation. During this phase, various resources such as journals, conference papers, articles are collected. The aim, purpose of study, objectives, and scopes are also provided and continued with the planning of research flow. Other than that, the dataset are also prepared during phase 1. In phase 2, which is classification, the methods to be used for classification are chosen and will be applied. Lastly, phase 3 is about prediction assessment, in which the result for each classification method will be evaluated, analyzed, compared, and discussed. The classification performance for each method will be measured based on accuracy, precision, and recall. In this part, the research design and implementation will be explained in detail through data preparation and model parameter. Data preparation is the method of cleaning and transformation of raw data before proceed to processing and analysis. In this study, the data is retrieved from GEO repository with an accession number GSE25507. The dataset is prepared by an experiment profiling by array in which microarray from gene expression were made on purified RNA from peripheral blood lymphocytes in children. Next, the dataset contained 54,613 features along with 146 samples. Quantile normalization is implemented in which it is used to change all sets of data. Machine learning models are parameterized so that the models can adjust their behaviors based on a given problem. For MLP, the parameters are the weight and neurons. Next, no parameter is needed for KNN is since it is a lazy algorithm. As for naive Bayes, the parameter depends on two types of features which are categorical and numerical. Lastly, the parameter used in SVM is to maximize the boundary between a given pair of classes. So here is the last part of this presentation, which is the conclusion. Throughout this project study, the first objective has been achieved, which is identifying classification method that can be implemented on autism gene expression data. Hence, this study can be a guideline in choosing the suitable methods for gene expression data classification in ASD. There are some limitations that have been encountered during this study. The classification methods used are more focused on the identification of gene expression data. Therefore, they may not be effective in detecting other types of dataset such as structural MRI or functional MRI data. There are some parts that can be improved for further research in the future, which is combining the current dataset used with another types of data so that a more useful result can be obtained. As for PSM2 execution plan, feature selection method can be applied to the machine learning methods that shows the best performance in terms of accuracy, precision, and recall. All in all, I hope that this research can give some insights and provide a useful guideline in the future. I think that's all from me. Thank you.